Godlewski. I work at the Department of Physiological Science of Faculty of Veterinary Medicine and Warsaw University of Life Sciences. And in my work, I usually work with nanoparticles. So I study the mechanism of interaction of nanoparticles with body, with the animal models, with the mouse models. And also one of my postdocs works on the cellular interaction and to some modeling of the interactions. Typical imaging task would be from the fixed samples would be just analysis, cytometric analysis of the distribution patterns and accumulation of the nanoparticles in the body. So this would be done on fixed specimens, but also we do live studies on the cellular cultures, and we also hoping very soon start to some experiments on the mouse, on the live mouse. But we still have to wait for nanoparticles that will be excitable into photon microscopy. When I started my PhD, we were looking for high definition imaging, so we were looking at different microscope, and we chose best optics that were available, so we went for Olympus systems at the time, and then we went through a lot of development. We went more into the cytometry, so more into the quantitative analysis. So we went through a lot of different systems, and we have now also one from Olympus system, the scanner system. When we switched our focus on the nanoparticles, we needed the different excitation emission spectra, and very, very difficult excitation emission spectra from the new fluorophores or new nanoparticles actually. And this made us to go for the white light laser, so we got very, very tunable excitation wavelengths. From the point of view of microscopy, the most important thing would be the resolution of the lenses and lack of the chromatic aberration, because we want to have the initial image, not process image, as close to the original as possible. So this is what we work mostly on. And this is also the only thing that we could do for cytometry, because then any post-processing will destroy the actual credibility of the data, so we always work on the raw data there. Lenses are the most important ones there. Sometimes with very difficult tissues with a lot of background signal, so we're looking into finding the fluorophores that will not interact with this, and also very narrow band filters, because then we can eliminate autofluorescence to a certain extent at least. And also spectral and mixing is very beneficial for this, and with the film techniques we can also eliminate a lot of background signal due to the differences in the lifetime of the fluorophore and the autofluorescence of the tissues. What ensures the proper preparation? Well, then we have certain pathways to follow or certain schematics of the procedure, but then expertise. So when we start with the students, they have to go through the whole process of hard learning of the techniques. The expertise is the most important, so try and try again, in other words. With the cells, we recently switched to live imaging, so generally what we need from the cell is the lack of the background noise, actually. So unfortunately, we had to modify very much the cell culture medium, because it was fluorescent. We had high fluorescence in the spectra that we usually measure the nanoparticles. And the conditions of the imaging, because we need to have them stable, and especially for the long-term observation of very delicate tissues like the neurons that we use, the primary neuronal cultures. This is of importance, the stability of the conditions, the maintenance of the stability. What I'm interested from the new system is the new FluoView 3000 system that you're presenting all around. And the specifics of the system would be this low magnification, high resolution imaging, so to image the whole tissue slice in one go, and also the spectral and mixing or spectral imaging that will allow me to get the best signal or tune to the best signal from the nanoparticles that I'm using. Right now we're running the Olympus reference laboratory, which encompasses a lot of imaging techniques from bright field morphometry and cytometry through fluorescent cytometry techniques, and also high resolution imaging from confocal system. And we like to, or we're going to improve it by acquiring this new FluoView 3000 system with the spectral imaging. and also one of the enclosed confocal system for live imaging of the bacteria and viral interaction with the tissues and cells.